All right, guys, let's finish this discussion on genetically modified organisms. Just a reminder, we actually learned what genetically modified organisms are. They are any organism that has been created, any organism that kind of has been changed in the terms of their genetics, so their DNA. It's typically thought of as plants or animals, but well, typically plants. We do genetically modify some animals, specifically if you go back into the traditional times, which we'll talk about in a second. Dog breeds were genetically modified, but even some salmon have been genetically modified to just kind of give them the traits that are desirable for either us or for eating, or desirable in terms of survival so that they can pass on their genes. We were going through some fact and fiction when my timer kind of cut me off, so I just wanted to make sure you are fully understanding this very last fact or fiction. So there aren't any known environmental risks to producing GMOs. Fact or fiction is actually fiction. So potential environmental risks, they're known, which leads to monitoring and testing before approval of new genetically modified crops. Scientists monitor GM crops to watch for unintended consequences that could be seen long-term, such as herbicide tolerance, biodiversity concerns, and effect on non-target organisms. So actually, we'll talk about it in a second, but we have been selectively breeding for crops, like the best traits that we want in our crops, and that has led to having a completely a crop base or a base for our seeds that's exactly the same genetics-wise. And if we get into studying populations really in depth, you're going to learn that the more genetically variant or diverse a population is, the better chance it has of surviving potential disasters, like either natural disasters or genetic disasters like diseases or predators. So the more diverse an environment or a population is, the better able it is to handle those kind of right? Those scary things, not scary things, those things that are going to be harmful to the population. So in the case of the Irish potato famine, I can't remember what year it happened in, but what had happened was a blight or a pest came and wiped out the entire crop of potatoes in Ireland back in the day. And that was not only their way to make money, these farmers, but it was also provided food for all of Ireland. And so people actually started starving. They were starved so much so that they would commit petty crimes just to be thrown in jail so that they could get 3 meals a day. So it has unintended consequences that sometimes scientists can't even predict. And actually that phenomenon of people committing crimes so that they can go to prison to get their basic needs met is something that we still see today. If we talk if we decide to take a gallivant into the humanities, that's actually one of the problems that we see here today, even in the US. All right, so plants that are genetically modified, I told you there was not any plants that are genetically modified. We went through that fact and fiction yesterday that most of the produce on the shelf is genetically modified. That's absolutely false. Here's the plants that are. Corn is our big one, and it's actually, we feed our crop animals like cows, genetically modified corn, but we have genetically modified soybeans, cotton, alfalfa, sugar beets, canola, squash/zucchinis, papayas, potatoes, and apples. That's about it. So if you see a carrot laying around, it's probably not genetically modified, although natural carrots are not orange like that, like you see. They come in all kinds of different colors. Fun fact. But anyways, that's besides the point. All right, so let's kind of go through a historic and present day perspective on genetic modification. So we've been doing natural genetic modification for years. We do it using crossbreeding, selective breeding, and selective pressure. So we humans have been messing with the genetics of organisms for many, many years. We do it in dog breeds, right? All the dogs originated from wild wolves, and then eventually we selected the traits we wanted for. Now we have all these different breeds. Horses, right? You want to breed a thoroughbred, like a really good horse with another really good horse, so that we have a great, so that we have a better chance of our offspring being great as well. And then selective pressure, so changing things so that nature or natural selection will select for those individuals. Crossbreeding, so these are your notes. produces an animal or plant by mating two different species, breeds, or varieties to create a hybrid. So in this case, we had some crossbreeding of what looks like cacti or succulents, not sure, don't quote me on that, but we wanted one that kind of looks in between. So we breeded the two, and we got the result on your right there. Now, crossbreeding is not super specific. You might not get the intended results. Your organism might not look like what you want, and it takes a while, right? So you have to wait for the plant to grow and to populate, and then if you wanted it even further to kind of become more of a hybrid, you'd have to wait for each generation. So it's pretty slow, but it is what we call a natural genetic modification, and we've been doing it since back in the day of Gregory Mendel and his pea plants. We have selective breeding, which involves choosing the parents with particular characteristics to breed together and produce offspring with more desirable traits. So if I would like, these look like bamboo to me, so we're going to call them bamboo. If I want bamboo that is nice and tall, I'm going to pick parent ones to breed. So pick two with your desired trait. I want them nice and tall. I'm going to breed those together so that my second population will have a better chance of having those desirable characteristics, being tall. And lastly, we have selective pressure, and that's when you change the environment of your species. So environmental factors or influences which may lead to less in reproduction in a species population and therefore contribute to evolutionary changes or even extinction through natural selection. So we talked a lot about bacteria and antibiotics. So let's say if we start on our left here, the population of bacteria with a subset of antibiotic-resistant organisms. So let's say that those bacteria, they're all just hanging out, doing their thing, bacteria thing. And then we enter in the middle page right there, middle picture, sorry, the presence of antibiotics. So those ones that were not resistant to antibiotics are going to die off, meaning that only the organism with the antibiotic-resistant gene is going to have the chance to multiply, and therefore its offspring are also going to, well, most likely, not all the time, but most likely have that antibiotic-resistant gene and become resistant to the bacteria or to the antibiotics as well. This is how we get what we call superbacteria, which is what we talked about way back when in our study on diseases. And lastly, let us talk for a second about transfection and CRISPR. So those three were what we call our historical genetic modification. Now we're going to talk about what science and technology, as they have progressed, what we have come to know as genetic modification today. So this first process we're going to talk about is transfection. So it's a process of deliberately introducing genes into an organism's genome. It's done using a plasmid, which is a small circular piece of DNA. It autonomously replicates, meaning it does it on its own. It's like a mini chromosome, so they're found in bacteria. They're bacterial DNA, but it has its normal DNA, but plasmids are just little mini ones. They're found naturally in bacteria, yeast, and plants, and can theoretically, so unless there are some constraining things that we don't know about, they can be transferred to anything with DNA, although it's not always possible. It doesn't always work out in theory like it should. So here's how it works. So with our, in normal life of a bacteria, when it can replicate just normally, where the bacterial DNA and the plasmid DNA are separate. They stay separate, it's just the same, so you're getting an exact same DNA makeup of the bacteria here and here as we have here. However, it can also replicate by incorporating the plasmid into its own DNA and then replicating again, creating an entirely new bacteria with that integrated plasmid. And the way that scientists use this is those plasmids can actually be taken out, selected for whatever trait you would like it to have, and then put into a DNA of something else. So commonly in labs, this is used, or I've used it in terms of making an organism glow. So bioluminescent organisms, they glow, like jellyfish and lightning bugs. If we took the DNA of a jellyfish, just that glowing gene, the GFP gene, and we inserted it into a plasmid, so this DNA that kind of moves freely, the circular, it's all by itself, it replicates by itself. We inserted that gene into a plasmid and then took the plasmid and put it into yeast cells. And after we put it into the yeast cell, and the yeast cell incorporated that plasmid with our new gene in it into its own DNA, then that yeast cell, which could not glow beforehand, was able to glow because it took up that GFP, or green fluorescent protein gene as well. So it's really cool, and scientists use it often in terms of, that was just for an identification, but it could be used theoretically to insert many genes, any gene, into anything with DNA. Not always possible, but it's what we're working with. That's usually how we genetically modify organisms right now. And lastly, I wanted to talk about Crispr. So Crispr is pretty exciting as far as scientific advancement. It's a family of DNA sequences found in genomes of prokaryotic organisms. What they do is they think of them like scissors. They allow researchers to easily alter the DNA sequence and modify gene functions of organisms. Anything with DNA, theoretically. And so this has been a major scientific advancement. It allows us to be able to take the traits that we want from one organism. So say I was trying to create, and we're going to do this, we're going to design our perfect predator in our next unit. Say I wanted to take this perfect predator and I wanted it to have the eyes of a hawk. So I would take the genes that give the hawk its good eyesight out of the hawk, put it in a use CRISPR to insert it maybe into my predator. Say maybe I wanted to create, I don't know, I want mine to be tetrapods, so I want it to have 4 limbs. So maybe I want the speed of a cheetah, so I put it in the cheetah's DNA. So now my cheetah has the speed of a cheetah because it's normally a cheetah, but it also has the eyes of the hawk because I've put the hawk DNA into the cheetah DNA. Okay, and you would have to do it as it was a baby and then grow it out of its cells. So don't think that I could take an adult cheetah and immediately give it hawk eyes. It doesn't work that way. You have to start from baby, embryo, very small, single cell, and then let it grow like we normally do. Okay, and then say maybe I also wanted my cheetah though to be camouflaged like a chameleon, to be able to change its color. That would be really hard with fur, but it's going to happen because that's what I want. So I'm going to take that DNA from a chameleon that causes it to be able to alter its pigment to fit its surroundings, and I'm going to put that in a cheetah. And so now I not only have a really fast organism, I have one that can camouflage, and I have one with the eyes of a hawk. Can you see how this is huge for science, but also huge for scientific imagination as well, how we can do a lot with this? It's really exciting and really cool. But this is the end of my lecture on CRISPR. I do have a whole video here. It is kind of long, but it's really interesting to see how CRISPR is changing the scientific biotechnology community as a whole. So I'm going to put this link as well as another link, as well as another link to GMOs on the page for Canvas so you can watch it too. Just watch it. You don't really have to comment on it. I really think it's going to help further your knowledge into this subject as well, though. All right, guys, have a great day.